Okay, the first MCQ that I got wrong was question number 3. Question number 3 read, researchers observe that the water within the high CO2 concentration has a lower pH than does the water with the low CO2 concentration. The researchers claim that lower pH alters the structure of the enzymes of photosynthesis. Which of the following most likely explains how a lower pH could alter the structure of photosynthetic enzymes? I said option B. The correct answer was option D. Option B said there will not be enough H plus ions for the necessary hydrogen bonds the context freaking option B said there will not be enough H plus ions for the necessary hydrogen bonds to form in the secondary, tertiary, and quaternary structure of the enzymes. This is so pH scale. I misinterpreted the pH scale. The correct answer D said there will be too much H plus ions, and it's in the question it says that there will be lower pH, which means it's more acidic, which means more pH. So I just misinterpreted the question and thought that there was less H plus ions, but actually there's more, which can still disrupt the number of the bonding. A is wrong because it says there will not be enough H plus ions for the ribosome to correctly form peptide bonds between amino acids. This is just wrong because the ribosome is not involved in protein folding, it's just involved in creating the proteins themselves. C is wrong because it says there will be additional H plus ions that can disrupt the peptide bonds in the primary structure. This is wrong because the denaturing is with folding, not the peptide bonds in the first one, because the first one is just how the protein is assembled, not the actual folding. Okay, next question I got wrong was question 4. Question 4 said, well, it was a big question, so for the following group of questions, first, okay, and there's like a graph thing. Which of the following, I wrote it all down to write in the script, but I don't think I actually need to write all that because it's just sort of like context, I guess. Anyway, the graph says that hydrogen peroxide solution goes from 1% to 5% to 10% to 15%, and then the average time in seconds for the discs to float goes from 30 to 25 to 20 to 10, respectively. And then 4 says, which of the following best describes why ice was used during this experiment? And the correct answer was B, and I said D. So B was correct because like ice makes it colder, which would slow down the reaction. B said that it would slow down the breakdown of the catalase, which is correct because ice makes it colder, which makes reactions slower to happen. My answer was D, to increase the size of the active site in the enzyme. Or no, E, sorry. My answer was D, to slow down the catalase molecules to increase the probability of contact with hydrogen peroxide molecules, but this doesn't make sense because ice would slow the reaction down, not increase it. A is wrong because increase the activity is wrong and would slow it down. C is wrong because purge the solution of excess air. That's not a goal that they have, and it's not relevant. I can't put down the code. D, E, increase the size of the active site. There's no correlation at all. Ice doesn't do that. I can't put down the cup. Six is the next one I got wrong. For 6, I said D. The correct answer actually ended up being A. So D said, the question said, really graph about fructose 6 and fructose 1.6. I did terrible in this question. It's happening. Phosphorylakinase is an enzyme that catalyzes the conversion of fructose 6-phosphate to fructose 1.6 biphosphate during glycolysis, as represented in Figure 1. PFK can be allosterically inhibited by ATP at high concentration. Which of the following is the benefit of regulating glycolysis by the concentration of ATP? Well, my answer D was glycolysis. Honestly, I didn't know at all for this question. I sort of just guessed. I say glycolysis is inhibited when the intracellular concentration of ATP is high because ATP will compete with fructose 1.6 for biphosphate for binding to the active site. I don't even know why I put that. The correct answer A, glycolysis proceeds when the intracellular concentration of ATP is low, which provides ATP to drive cellular reactions. Yeah, and this makes sense because glycolysis produces ATP, so if there's low ATP, glycolysis will happen so that there's more ATP produced because the body needs ATP energy. Anyway, question B, answer B is wrong. Glycolysis proceeds when the intracellular concentration of ATP is high and the cell stores ATP for future use. This does make sense because why would it do more when there's a higher concentration of ATP? It doesn't make sense. If there's already so much, it might focus on other stuff. C. Glycolysis is inhibited when the intracellular concentration of ATP is low because PFK requires ATP as a substrate for the reaction it catalyzes. This is wrong because it's saying that they compete with each other a little bit. It's like, it's just multiple other cellular reactions, not just the PFK one. I can't put down the cup. Okay. Next question I got wrong. Oh my god, my hair is so freaked out. I said my answer was B. The correct answer was dude, I think I organized this so poorly. Correct answer was C. Okay. Student places 20 tobacco seeds of the same species on moist paper towels in each of 2 Petri dishes. Dish A was warped complete. I think I misread that. Dish A was warped completely in an opaque cover. Oh, dish A was wrapped completely in an opaque cover to exude all light. Dish B was not wrapped. The dishes were placed equidistant from a light source to a set of cycle 14 hours and 10 hours of dark. All other conditions were placed equidistant. All other conditions were the same for both dishes. The dishes were examined after 7 days, and the opaque cover was permanently removed from the dish. Both dishes were returned to the light and examined again at 14 days. The following data was obtained. And then it's a data about, like, germinated seeds, the seedlings, how many of them are yellow, stuff like that. And then, so I'll get to go. My incorrect answer was, I said B. Well, the question was, additional observations were made on day 21, and no yellow leaf seedlings were found alive in either dish. This is mostly because I said taller green leaf seedlings blocked the light and prevented photosynthesis. Oh my god. And prevented photosynthesis. How am I doing, bro? This is wrong because they would still grow, they would just be inhibited, so they'd probably still be yellow. The correct answer was C. Yellow-leaded seedlings. Oh my god. Were unable to convert light energy to chemical energy. This makes sense because that means that they did not have the correct pigments to convert the light energy. A is wrong. Yellow-leaf seedlings weren't able to absorb water from the paper towels. This is wrong because the water is not direct correlation with energy conversions. Option D, higher rate of respiration in yellow leaf seedlings depleted their stored nutrients. Doesn't make sense. Higher respiration would just mean more of doing the energy, which would probably make me even greener, honestly. If I had to guess. Where, I hear your voice. No, bro. I don't even know what I'm gonna do for lunch tomorrow. Okay, Miss Fence. We soldiered through it. Now why don't you just let us take pictures, bro? It was so much easier than writing everything down. Oh God, sometimes I write it down. It would be so much easier. Literally, save me so much effort and time. Sipping on promethazine. My hair is freaked up. Anyway. Next, I got question 16 wrong. In 16, I said A. Meanwhile, the correct answer was C. So, it's a diagram of okay. 16 has a diagram of electron transport chain. Is what I wrote. I don't know what that means anymore. I don't remember. I do not recall. But apparently it had a diagram there. It says compound X binds to compound 4. Wait. Okay, this is weird. But I assume it was written in Roman numerals because I kept that. I said compound X binds to complex. Compound X binds to complex 4, but I wrote it in Roman numerals. Maybe it's compound X, compound 10. A compound 10 binds to compound 4, or complex 4, of the mitochondrial electron transport chain and prevents complex 4 from acquiring electrons. Based on figure 1, which of the following best explains why the cells of an animal exposed to compound 10, yeah, whatever. Compound 10 have an increased ratio of NADH to NAD+. I said NADH increases because compound X directly prevents ATP synthase from producing the ATP necessary for the evolution of ATP of NADH to NAD+. So, this is wrong. I said this was wrong. Or no, I said this was right when I was taking the test. The reason I said it was right was because I thought that oh my God. Okay. The reason I said that A was right was because I thought that compound X binded to the ATP synthase, which would stop ATP production and therefore stop the whole process. However, this is wrong because compound X does not do that. It does not bind to the ATP synthase. This could be attributed to a different thing. B. NADH increases because the binding of compound X to compound 4 prevents the transport of NAD+ out of the mitochondria. It's just not true. This is if you just look at the graph. I wrote one. Okay. How to say if you look at the graph, it's pretty simple, but I don't have the graph with me anymore. Because I can't put down the good. Option C. NADH can oxidize to NAD+ because complexes 1, 2, 3 cannot accept electrons if electrons cannot be passed to complex 4. This is correct because complex 4 becomes first. I said in the diagram it comes first, and so it cannot pass the other ones. Option D. NADH cannot be oxidized to NAD+ because H2O cannot bind the active site of complex 4. if the complex cannot accept electrons. This is not true, because H2O could bind anyway. Okay. Final home stretch. The last question on the multiple choice before I end the video and start a new one for free response. Okay, question 20. Question 20, I said A. The correct answer was D. Question 20. The enzyme hexokinase catalyzes conversion of glucose to glucose 6-phosphate, which is an important step in glycolysis. The reaction involves the transfer of phosphate group from ATP to glucose. Either glucose molecule or water molecule can fit in the active site in hexokinase. The presence of a water molecule in hexokinase active site would result in the hydrolysis of ATP to ADP instead of the conversion of glucose to glucose 6-phosphate. Which of the following statements best helps explain the reaction specificity of hexokinase? Again, I said A. Both glucose and water are polar molecules that form favorable interaction with charged and polar amino acid side chains, and hexokinase is actually. I really had no idea what this question was asking. I think I picked this because I thought it was true, but I don't really know how that actually talks about reaction specificity at all, so that's my reasoning. Option D was the correct one. Glucose has the right shape and charge to cause hexokinase to undergo a structural change needed for catalyst, while water is not. This just makes sense because, like obviously, I thought it was too simple. And when I was taking the test, I took a nap before this, by the way. I was such a dry my hair, so freaked out and like going every which way. Oh, good. Okay. This makes sense because that's what reaction specificity is that it would be specific to the glucose and not the water. Whereas mine is saying the opposite, that it would sort of bind to both when we know that's not true. Both glucose and water, option B, both glucose and water have oxygen atoms that can form covalent bonds with the phosphate atoms of phosphate groups. Glucose is an energy, oh, okay, sorry, I was reading the next question instead of my explanation. Also, option B is wrong for the same reason that option A is wrong, because that's just like saying it's the same thing compared to reaction specificity would mean different thing, like specific for the glucose that the water did not have. I can't put down the cup. Option C, glucose is an energy-rich organic molecule that can be broken down by glycolysis to produce ATP, whereas water is an organic molecule. This is just like not relevant at all, I don't think. Anyway, that is my end of my multiple choice. Bro, my hair is so freaked up. And I cannot put down the cup. Wait, pop quiz, Miss Vance. Do you know everyone in our AB Bio class is dating? Pop quiz. Okay, fine. Now FRQ, and after I take a quick snack break.